So first of all, what we're here for, we are launching the new Office of Clinical Trial Development and Management. And this is really a milestone for all of us. I know that it's part of, it's our commitment to grow clinical trials. We are all about improving patient care here across Western New York, and bringing state-of-the-art research to patients is critical. To do that, we want to have the best resources possible, and that means announcing the establishment of the new clinical trial statistical and data management program. This is under the leadership of Dr. Greg Wilding that we just were mentioning. It's a centralized hub that will provide essential statistical and data management support to UB investigators. So by fostering this interdisciplinary collaboration, we aim to enhance planning and execution of large-scale clinical trials, and ultimately this enhances our research endeavors and gets us to our goal of the top 25. Dr. Wilding, I know, not sure how many of you know him, but he's a professor at UB's Department of Biostatistics at the School of Public Health and Health Professions. He has over 20 years of experience in diverse areas of clinical research in both academic endeavors as well as industry. He's been a pivotal biostatistical investigator on numerous funded clinical trials. His expertise spans clinical trial design, permutation tests, resampling techniques, goodness fit tests, distributional characterization, and many, many, many more. His academic journey started with a PhD and a master's in statistics at the University of Rochester, just down the street. And he has a Bachelor of Science in Mathematics and Business Administration from SUNY at Brockport. He's had significant careers, including having roles not only here at UB, but also at the Roswell Park Comprehensive Cancer Center. We're really confident in Dr. Wilding's ability to set this up. I do see that this is a cornerstone of our research community. It really promotes rigorous and innovative clinical trials. I see Dr. Murphy here on the screen, and he was one of the big champions for going ahead and doing this. It lays the groundwork so that we can have new investigators, which is near and dear to all of our hearts, develop innovative investigator-initiated trials to make sure that they have the preliminary data that is needed to advance to getting big submissions like PCORI, NIH, or to take something to industry for those type of innovations. I also want to acknowledge a newest team member, Austin Glick. He's a clinical data manager, and it's great to have Austin on board. Thank you to everyone for joining us here today. And thank you to Dean Watuski-Wendy for her support, Dr. Murphy for her support. It was through the loud cries from the deans and Dr. Murphy that we established this. And again, I want to thank Dr. Cizek for her vision and perseverance in getting this set up. I think it will lay the groundwork for many, many future endeavors. So thank you so much. Really appreciate everyone's work.
Thank you, Dr. Brashear, for making the time to join us. So Greg, with that said, I think we hand it off to you, and I will spotlight you, and I'm assuming that there you go. You're already sharing slides. All right. Thanks, Greg.
Thank you very much. So I'm Greg Wilding. As Dr. Brashear said, I'm in the Department of Biostatistics. I've actually been here for a while. I've been here since 2002. It was my first gig out of grad school. So I came here at a time when there was really very little biostatistics across campus, and I've been lucky enough to be part of the growth of biostatistical endeavors over the past 2 decades and some. So why did we start this new office? A lot goes into planning a trial, and what I'm going to focus on here is stuff that I have expertise in. There are challenges outside of statistics and data management, but when I think about the sort of things that I would have to worry about as a clinical trialist in this area, you have to worry about picking the appropriate study design. You have to worry about designing your database properly, implementing proper data collection procedures. You're always worried about the quality and the integrity of your data. A lot of times during studies, we might do an interim analysis, and it might involve halting the study temporarily. And so we have to kind of think about that. How can we do that in as quick as a way as possible? Safety, making sure that we're protecting our patients, our participants, using valid and efficient statistical methods, and things like data sharing and reporting. Traditionally, UB has not had a centralized mechanism to handle this. If I wanted to conduct a very large, important clinical trial as an investigator, what I would have to do here at UB, say a year ago, is I would have to start looking for resources around campus. And this could be a huge burden on our PIs. A lot of times you could find resources at a very small research group level, or you might talk to someone in their lab. They might have someone with the expertise, or you might have to use less than qualified individuals. For instance, maybe you might have to use your nurse coordinator also as your data manager. And they may have the expertise to do that, but often they don't. Or the other model is to use external organizations. And that can work really well. That has worked really well in the past. But a lot of times it has not worked well because a lot of times these external organizations are not as stable, or they don't necessarily prioritize academic projects. And so these are all large barriers to us writing very important clinical trial oriented grants. And so after a lot of planning, we started looking on the outside and look to see what our peer institutes would do and what institutes that we aspire to be did when they grew their own resources. And here's just a small subset of the sort of resources you can find around the country at different universities. There are a small collection of papers where people wrote about their experience in growing a unit that you see on the far right. And then, talking to a lot of my colleagues and reading these papers, you quickly see that really these are all different environments. They all had their own challenges. The way that they approached it was different, but there was this common theme, right? There's two common themes that at least I picked up on. One is you capitalize on your existing resources. So you look around campus, you see what you have, and you use that as the base to grow something, and then you invest in that growth. And so that's what we did here when we started this new office this past summer, which I think is April when we first started working on this. The Office of Clinical Trial Development and Implementation, this builds on two very strong units that we already have here at UB. It builds on the Department of Biostatistics. The Department of Biostatistics, the formation of that's actually why I came to UB many years ago. That was part of the university's plan to develop a school of public health and health professions. So we became a department within that school. We have, of course, our own academic mission of conducting really strong methodological research. We do a lot with interdisciplinary research across really all the schools on campus. And we have our own educational programs that we support all the way from undergrad to doctorate. Our faculty have joint appointments with all the health science schools, and that really kind of goes to show you that really as a department, we have our finger on the pulse in terms of research here on campus, and we're heavily involved in that. And then we also have faculty within the AI and Data Science Institute here on campus. So that's the first unit. The second unit is our BIRD. And so if you're not familiar with the BIRD, that is a core within our Clinical and Translational Science Institute. BIRD stands for Biostatistics, Epidemiology, and Research Design. Most CTSIs have something like a BIRD. Ours is a little bit different in the sense that we span 2 institutes and 3 departments. So here at UB, it spans not only the Department of Biostatistics, but also the Department of Epidemiology and Environmental Health, where we get a lot of strong methodological support. And then also at Roswell Park, they have a Department of Biostatistics and Bioinformatics, and we have faculty within the BIRD that support that as well. And so these 2 units, they had a heavy role in terms of planning this new office that we've opened. So we're building on those units, and then we needed that investment, and that came from the VPHS office, where an investment was made for a clinical data manager, a senior statistician, and some funds that we're going to be using for students. And so all the people involved in the planning across many schools here on campus, we're all committed to helping grow this, because this is really key to our research mission here at UB in our aspiration to get into top 25. So our mission within the new office, we view this as a centralized hub for integrated statistical and data management infrastructure. The mission is to improve the planning and conduct of clinical trials by providing this high quality support. And then also it's not enough to just provide support. We actually also have to train our workforce, right? So we have a responsibility to make our faculty, our researchers here at UB better clinical trialists and introduce perhaps faculty that are interested in clinical trials to the world of clinical trials. We have 3 goals at this point. One is really about building infrastructure, and I'll go through some of the activities a little bit later on associated with these goals. But the first one is building this infrastructure, right? If we're going to get these large grants, we need to show that we have the infrastructure, we have the support here to support these large endeavors. The second is to actually support individual trials all the way from design to reporting. And the third is to provide skill building opportunities, not only for novices, new people that are new to clinical trials, but also our established researchers as well. This office will better position UB to go for this external funding. It will enhance our reputation here at UB, and really not only UB, but the SUNY system as a whole, because SUNY does not have anything like this here, like what we're growing here at UB in Buffalo. This is the personnel. So myself, I've worked in trials for a large number of years, both in academia and in industry. I've probably been involved, I say 200 plus trials. It's probably over 250 at this point, in many different phases, really all phases of intervention evaluation, and a lot of disease areas as well, a lot of different clinical areas. Most recently, we hired Austin Glick. I think it was April we hired Austin. He has a large amount of experience supporting large multi-center NIH clinical trials across many scientific areas. We also have two students in the office right now that are working on specific projects. And then hopefully by the end of the month or maybe the beginning of next month, we'll have a new senior statistician on board. I've made an unofficial offer. There's unofficial acceptance. We're just at this point just waiting for the official paperwork. So this is the office currently. We obviously expect it to grow over the years, but given an office's size and the really ambitious research mission of UB, we need to take advantage of some really key university partners. A partner for us, of course, would be the Department of Biostatistics. We're going to have a lot of expertise from the faculty there. These are faculty that would perhaps serve the role of study statistician with a lot of the clinical trials that we'll launch here at UB. Another partner would be the CTSI. With the CTSI, again, we have the bird that's located in CTSI, but we have a lot of other very strong cores that can assist us in building the team that's appropriate for your clinical trial. And then also it helps us with access to resources. On campus, if you're not familiar, there's something called Research Information System, which is in the VPR office. They're our hub right now for REDCap support, and there's also some database programmers there. And so those are individuals that we're working with now. And then of course, a natural partner for us would be the clinical research office as well. Now, because of our expertise within the office and our partners, we can handle pretty much any clinical trial that you might throw at us. One of the challenges of clinical trials is depending on the clinical area you're working in and the phase of evaluation you're in, you can have very different looking designs and analysis methods and data management challenges. Luckily, we have experience in many realms. So when I think about how I might classify a trial and the sort of key features, we have a lot of experience in evaluating drugs, medical devices, preventions, maybe a prevention intervention for disease, procedure such as a surgical procedure, behavioral interventions that maybe you might use with a patient population suffering from addictions, educational interventions, diagnostic tools, and delivery systems. These are all things that we have experience in. We've done things in early phase. There's a lot of challenges when you first embark on that first clinical trial because you're most likely dealing with very limited resources. And so you might conduct a pilot study, or you might conduct a phase 1 or phase 2 trial. If this is in the drug world, there's specific features that you'll only see in a phase 1 that you would not see anywhere else. And these are things that we have expertise in. Dose finding, if dose is appropriate for your intervention, proof of concept study, where we're trying to see, do you have any kind of signal? And then also late phase trials where we do confirmatory trials. These are more like your phase 3, phase 4 trials. Analyses to establish superiority, non-inferiority, or equivalence. We're well versed in single and multi-site trials. Obviously, with multi-site trials, there's a lot of complications that you have to think about during the planning phase and during, of course, during the implementation. We have expertise in parallel group, crossover, factorial, step wedge. We do a lot of step wedge trials nowadays. Cluster randomized, individual randomized group treatment trials, which a lot of people here do on campus, but they don't realize they're doing it. It's when you randomize individuals, but you treat them within groups. And there's very unique statistical features to that data structure that need to be taken into account. Adaptive designs, we have a lot of expertise in that. If you're dealing with a very, maybe serious intervention, or you're dealing with a vulnerable patient population, you might want to do an analysis halfway and see if maybe you should call it quits on the trial if the intervention is not doing what you think it would do. And so you need to be very careful on how you plan that. And then, of course, we do a lot of explanatory trials where you're conducting the science under an ideal condition, but also a lot of pragmatic trials that we've been involved in. On the right is a diagram kind of showing the different areas where we might help you in terms of study design, randomization, analyses, programming, a lot on the electronic data capture system side, making sure that your data is of really good quality, and then, of course, helping with any kind of reporting needs that you might have. So to kind of get in some of what we're doing right now, it's been a really busy summer for myself and Austin and the students. What we've been doing right now is a lot of infrastructure creation, right? So if we're going to apply to these big grants, we need to talk about what we've built here, right? Because that's what our competition has, right? This is not any kind of crazy idea that I came up with. I'm just copying what our competition has, right? So right now we're working on a lot of SOPs and workflows. We're developing a software library because if you're conducting these really large clinical trials, you got to make sure that you have your output in a very standardized and efficient way. We're working on, a CRF library, making sure we have validated forms. We're making sure that we're versed in the EDCs here that we have access to on campus, such as REDCap and Encore. We're working a lot on template creation. This is really important as we go for grants that might have a pretty short window in terms of when we need to submit it. With large clinical trials, you often write a separate statistical analysis plan, data management plan. You might have a clinical study report as well that is kind of the basis for the publication that you might write. And then also with trials involving serious interventions or vulnerable patients, it's probably a good idea to have a data safety monitoring board. And so we help with the organization of that in terms of charters, member agreements, making sure that the analyses get turned around with the appropriate timeline. So with this creating infrastructure, this will better position us to go for those really large grants or centers that have a large clinical trial component to them, because we're going to be providing this comprehensive scientific support, not only to our PIs here for their investigator-initiated trials, but also we can help be a better industry partner as well for those that work with industry. Of course, with everything that we create here, we have emphasis on sustainability and also integrating with the already existing important units here at the university. So this is an example of what we might do if you are someone that hasn't worked with statisticians a lot, or maybe someone that hasn't even done a trial yet. This is the sort of team that we can kind of put together for you here on the lower left. That's kind of an example support team. This would be something that you would see for a rather large trial. So not only would you have a study statistician, but you're going to have an entire statistical team that's going to be working again from the planning of the trial all the way to reporting. And then you're going to have a data management team. And this team, this can interact with a lot of the study personnel where the burden would usually have fallen on the PI. So we can interact with people entering the data. We can interact with the people that are helping with the EDC. Of course, obviously, we interact with the investigational team, specifically the PI. And then again, if this is a trial that requires a data safety monitoring board, and this is supposed to someone that would operate independently of the investigative team, we would support them with the analyses that they require. On the right, you'll see a table of the sort of example support we would provide for a trial. I won't go through every element on here. I don't think we have the time, and I don't think you necessarily need to see that. But one thing I do want to point out here is on the far left column is that we're heavily involved in the design, right? We're going to help you design the trial. not only from the statistical design, but will help you with the randomization, will help you write that protocol or that grant application, will help you foresee any kind of data management challenges that you're going to have and help kind of write up the plan for that. In the event that we get that funding, fingers crossed, right? At startup, we help set up the randomization. We actually start programming that. And so for those that are not kind of used to this process, that might be surprising because like, why would you start programming when you don't have the data? Well, what we want to do is we want to report as soon as possible. So what we'll have by the time the database is locked is we'll have the analysis programs pretty much programmed and QC'd. And so that allows us to turn around the analysis output in a very timely fashion, right? And that gets up the results out to that population that need it. And so during that startup, we're starting to program, we're starting to prepare documentation and a lot of our processes. During the execution, we're helping you model the accrual, again, more programming, helping you with any kind of interim analyses if that's something that's appropriate for your trial. We're resolving any kind of queries that might take place. There might be some data entry that might take place. We're making sure that we're audit ready at that point. And then at closeout, we're producing that clinical study report. We're performing those analyses. We're helping you with reporting. And so not only would that include papers and abstracts, but that also might include clinicaltrials.gov, which I know a lot of PIs struggle with in terms of reporting to that and having the output in the right form as they require. And then, of course, we help with sharing. Depending on the mechanism that's funding the trial, you might have certain requirements in terms of sharing. And so we get you prepped for that in terms of having the data in the right form and helping you with the documentation. So those are the sort of example things that we can do for you. I'll spend only a few seconds on this slide because you might say, well, how much do we have to worry about this in the budget? Well, if it's a big, important trial, it is going to be a substantial amount of effort for the people involved. So these are some effort recommendations. These are actually not numbers I came up with. These are numbers that are generally what you'll see out there when you look at other similar units. And it really depends on your project. You see with a lot of these percentages, I'm giving you a really wide range because some projects are going to require a lot of work depending on the science involved and some not so much. Um, our focus of our office is on these larger projects, right? But really we view ourselves as a hub for all clinical trial research. So even if I cannot collaborate with you personally and my team can't collaborate with you, we can definitely refer you to someone here at UB that can help you, be it the Department of Biostatistics or the Bird Corps or any other unit. And so you can see that what we try to do is we try to at least give advice on any size trial at any point where you are during the evaluation process. What I'm going to do here is I'm going to take you through a workflow. This is not normally what you would see in a talk like this, but like one thing that I do encounter when I'm working with investigators is they don't realize the number of statistical decisions that need to be made. And these are not decisions that I would make by myself as a statistician. So these are things that everyone on the investigative team has to think about. This is actually still a work in process. I kind of looked for something like this before and I couldn't find it. So I made one on my own here, right? So, first, when you start planning a trial, you need to make sure that you have a standardized intervention. And that's something we can help you with, or that might be something that you've already figured out for yourself. But at that point, we can start thinking about this trial. And I find these are decisions that you want to make in conjunction with each other, right? So as I'm thinking about the objectives of the trial, what are the big questions I want to answer? I'm also going to think about what is the patient population or the subject population that this applies to. So that's why I think you can't think about these as two separate components. You got to think about them simultaneously. And that most likely will involve a control group. What's the appropriate control group here? Is it a control group that receives no treatment? Is it a group that receives placebo or standard of care? And that will be relevant also in terms of the sort of blinding that I can start to implement. Once I've done this, I can start thinking about what are my design options, right? So I could consider maybe a parallel group design or a crossover trial or a step wedge or a cluster randomized trial. These are the sort of things I can start to think about at this point. I'm also going to start to think about outcomes and the relevant estimates. If you're not familiar with that term, and I won't get too much into it, it's this idea that during the trial you might experience, or a subject might experience, these intercurrent events. For instance, a subject is given an assigned treatment, but they take an off protocol treatment as well, or they go missing, right? So I need to think about how I'm going to treat that into the analysis. And that's not solely a statistical decision. That has a lot of clinical input. And so these are things that you would also have to think about if you are the clinical PI. And then I'm going to start to think about sample size constraints. So once I've done that, and of course, if there's a hypothesis that's involved, I'm going to want to consider any kind of minimally clinically important differences. I'm going to start to ballpark the sample size, right? And so why am I saying ballpark? Well, at this point, I'm considering a number of potential primary outcomes. Also, I might consider down the line some adaptive components. I might consider some stratification. Maybe I'm not sure at this point. So this could be the final sample size I'm computing, or at least it's getting me in the right neighborhood. At that point, we assess, is this possible? If this is not possible, if I'm coming up with a sample size that's too large, I might have to go back to the drawing board, right? But if I am in a situation where this, okay, is a doable trial, I'm going to start to commit to what my primary and secondary outcomes are. At that point, I'm going to start to think about things like stratification. Now that I know what my primary outcome is there a good chance I might see severe imbalance at baseline just due to chance? And so I'm going to start to think about what are those stratification variables that I want to have balanced at baseline. And that's connected directly to the randomization method I might use. I'm going to have a lot of choices for randomization. All have different pros and cons. Once I'm at this point, now I start to get into the nitty gritty analysis methods, right? So I'll come up with my main analysis is, I might have to implement some multiple testing strategies. If you've taken an intro to stat, you probably remember Bonferroni. If I'm going to compare three groups to each other, I want to correct for a false positive result. In trials, you might use that as well. But in trials, there's a lot of specialized methods for multiple testing. You might have multiple primary outcomes, and they might not all be equal in terms of their clinical importance. You might have an established hierarchy. And so there are methods in statistics that are designed exactly for that particular scenario. I might start to think about adaptive components to my design. Do I want to possibly stop early? Do I want to maybe re-estimate my sample size halfway through? Do I want to add an arm or drop an arm based on what I see in the data? And that might lead to a refinement of my sample size. And then finally, I start thinking about how I treat missing values. I might think about sensitivity analyses, because a lot of my methods are going to make assumptions which may not be true. And so I want to evaluate the robustness of my result if I start to consider other assumptions. Supplementary analyses, subgroup analyses. These are things you're going to want to think of. And you're also going to be you want to be very aware of what the funder requires. And so, for instance, the office recently helped with a couple PCORI submissions a couple weeks ago. And, regardless of how we feel, these are things that PCORI wanted to see. And so you need to be very familiar with what is required by that funding agency. And these are things that we can help you understand and help you address within the office. At this point, I'm going to turn it over to Austin Glick, who's going to talk a little bit about our data management process.
All right, thanks, Greg. Hello, everyone. Like Greg said, my name is Austin Glick. I am a clinical data manager here in the Office of Clinical Trials Development and Implementation. And I'll be giving a quick rundown of what the data management workflow would look like for the office itself. So that's going to start with protocol review and development. Now, the data manager doesn't have a very in-depth role in the early phase of development, but their workflow does start during the protocol review. Their data manager is going to be assisting the study team during the development process, and they're going to weigh in on any data-related questions or topics of discussion. And they're also do tend to assist with reviews, sit on planning team calls, and they're going to start taking some considerations into what data management aspects the study is going to need as we move forward. So once the protocol's been reviewed and developed a little bit, and we're starting to move into the next phase, which is where we need to identify our data collection needs. And now this is where the data manager takes a really upfront role in this. So from there, one of the most important steps during early protocol development is identifying what types of data collection elements need to be included in the study. And this could include anything from case report forms, either electronic or paper, a randomization system, any type of reports that are required either at the sponsor level or at the study team level, setting up a electronic patient-reported outcomes system, or ePro, electronic kind of questionnaires, and then any other specific utilities that are needed for that protocol. So a lot of protocols differ. There can be some complexities that are required and some things that would shift how we tackle the data management needs of that study. So once we identify those initial data collection needs, then we need to go into our next step, which is identifying the electronic data capture system. And this is going to be utilized for the study. It's really important to implement. It's going to be there for the entire life cycle, start to finish, and it's going to define how our data is collected, what everything looks like, how it interfaces with the sites, potentially how it interfaces with the participants, if there's any questionnaires on there. So this is a really important element. And there's a wide range of EDC systems available, and each has its strengths and its weaknesses. So smaller local studies could be built in a system like REDCap, but REDCap has some challenges for use on large multi-center protocols. For these kind of studies, the data manager will work with the team to discuss any options for other larger EDC systems, things like Encore's EDC, OpenClinica, Metadata Rave, VDoc, just to name a few. And then they will go through the benefits of the certain systems, such as the capabilities, the ease of use, the reporting capabilities, and available add-on utilities, such as randomization. And then they'll also discuss the regulatory considerations for each system. You know, most cloud-based EDCs are inherently 21 CFR Part 11 compliant, but a system like REDCap needs to be configured locally to be compliant, which is a large consideration that needs to be made and something that's really important to hash out while we're in the early development phase. So then from there, we move down to any additional utility creation that needs to occur. So based on the EDC system we select, we would then identify with the team what additional utilities are available, and from those, what the team would like to use. So for example, if a study is multi-center, has a complex randomization structure, the data manager, along with the statistician, could present on implementing an online randomization system that could make things a little bit easier. And there also may be need for electronic questionnaires to be available to be completed by the participant. So the data manager may work on implementing an electronic patient-reported outcomes, or ePro system, also referred to as eCOA. And then from there, we get into one of the most important aspects of a data manager's job during the early phase of development, and that's creating the case report forms. So this step will vary greatly based on the study. The data manager is going to take the protocol, and they're going to develop a dynamic, rigorous set of forms that will follow good clinical data management practices, or GCDMP. And then these forms may also be using some data standards set by the Clinical Data Interchange Standards Consortium, also known as CDISC. And these standards, often referred to as C-, collect the data in a user-friendly, EDC CRF-friendly way that maximizes data quality and flows smoothly into the study data tabulation model, or SDTM. And SDTM is the organization and structure and format of a standard clinical study for the data sets. And it's often required by studies that need to report to the FDA. And all of these big acronyms really all just add up to a set of standards that lead to the best method and the best creation of CRFs, so that we have standard data at the end of the day that can be shared, used for review, and then reused by others if they want to take a look at it. So it's very important that when we're developing these CRFs, that we're following a lot of these standards and creating the forms in the best possible way. And then based off of the creation of the eCRFs, we have to then move to creating the database and getting the data set structure set up. To do this, we need to define the data mappings. And this is also one of those steps that's going to vary greatly from study to study. Smaller studies will require less complicated structures to be put into place. But in contrast, larger studies that require submission to the FDA will need SDTM and analysis data model, or ADAM, data sets to be set up. So most studies, regardless of the reporting requirements, will be set up within an Oracle database to ensure data safety and integrity. But this step is also going to vary greatly based on the complexity of the protocol. So these two steps, CRF design and the database setup, will vary in length and intensity based on how complicated the protocol is, how many treatment arms, how many participants, things like that. And that is going to be a really important step during study development. And then once the ECRFs are created, the database is set up, and then all the utilities are put into place, testing and validation can occur. And this is going to occur for each data element created during the study build process. And it will also occur throughout the life cycle of the study if any changes are ever made. So ECRFs will undergo intense user acceptance testing and validation to ensure no mistakes were made during the build process and that all forms flow properly. And data manager will be conducting a lot of their own testing during the development process. We usually call it chaos testing kind of in the industry. And that is to just put the forms through rigorous testing from different angles to make sure that we identify any possible issues that could occur. And the testing team will consist of individuals with a broad range of expertise and experience to ensure that all of our data elements are designed per our best practices. So we're going to have a lot of people who are interested in QA side of things. We'll have data managers, we'll have statisticians, we'll pull some site personnel in there as well to weigh in on some of the forms and development so that we make sure we're tackling it from all angles and that the forms are developed as best as possible. And then the end of the validation process will consist of sign-offs from both the build team and the study PIs. So then from here, once that's all completed, then we can move to study opening and we can start accruing. So now we're going to move into the second phase of the data management workflow, and that's the data review and validation, the phase that occurs throughout the life cycle of the study until closure. So while the study is open, data review and validation will be consistently occurring to ensure the best data integrity. These processes will vary based on the complexity of the study, like a lot of other things, but they'll be well defined within the data management plan of the study, which will be created way in the beginning. One part of data review that is going to occur is source data verification. This may or may not be done by the data management team based on the study. Large multi-center studies typically utilize study monitors or clinical research associates, also known as CRAs, that perform targeted source data verification and act as an independent entity. Studies where the data is collected at UB, we can leverage UB personnel and our data management team to perform source data verification. So that may or may not occur. But regardless of how it is performed, SDV will help to identify any issues with the study data and set up future data review processes, which is why it goes back into our data review and validation, because that's going to be affected heavily by the SDV. And then the other big aspect for a data manager is data querying and reports. So while data review is ongoing, data querying is going to be occurring consistently. Data querying based on data review findings will occur throughout the life cycle of the study, and the data manager and data manager assistant will utilize data review best practices, and they'll integrate a risk-based approach for data review to ensure the highest quality data is presented in the final data sets. So reports will also be simultaneously run throughout the study to present the study team with insights into the study's data integrity and the current accrual rates as well. And then reports can also be customized based on the needs of the protocol and the study team. So if a study team wants to see what sites are being queried the most, if it's a multi-center trial, and kind of create a metrics report to show what sites are accruing properly and what sites are falling a little bit behind, that can be done. And those reports are going to be developed during the life cycle of the study. Some may be developed early on based on the requests, but a lot of them are going to be created during this process. And then the other thing that connects to data querying is data corrections. So any data issues that are identified for significant data points, things like participant ID issues, date of birth being incorrect, treatment arms being assigned incorrectly, etc. These will be corrected using a set data correction process. And this process allows for proper documentation of important changes and can be presented to key personnel in the case of an audit. And data corrections may not need to occur for a study, but the process will be set up for each study in case they ever do. So we hope we never have to do a data correction, but in the reality of things, mistakes can occur. And the data correction process is there for significant issues. Any minor data and misreported lab value, things like that, won't require a data correction. But anything significant, like a date of birth being reported incorrectly, that would require this process. And we hope to never use it, but we always will implement it just in case. So now, after all this is done, all the reviewing is continuously occurring, and the study reaches accrual or it's prematurely closed for any reason, we move on to the next kind of phase for data management and for the study, which is study closure. So we'll move to a database lock, and during a database lock, the data will undergo a thorough review and cleaning, and all outstanding data queries will be addressed, and data validation will once again occur. It's going to occur throughout the life cycle of the study, but we're going to make sure that we're reviewing all of the data as we get ready for a lock. And then once the database is sufficiently cleaned and no issues remain, the database will be locked for data entry and changes. So this way, the sites won't be able to make any more additional changes. And then we'll also send them for signatures to make sure all the data is signed off on per 21 CFR Part 11. And then we'll move into the last part, which is the study analyses. So once the database is locked, then the study analysis can occur from the pre-established data sets that were defined at the beginning of the study development phase. And this is kind of where the workflow ends for a data manager. But this is what a standard data management workflow could look like. However, I do want to preface it and state that every study is unique. So certain elements of the workflow may need to be adapted to meet the needs of the protocol and the study team. Certain complex studies will have additional reviews, additional things that need to occur, and some smaller studies, we might be able to skip a few of these steps just to move forward with the study a little bit quicker. But that was kind of an idea of what a data management workflow would look like if you were to work with the office.
So obviously, by having such a unique unit here at Buffalo, and the fact that, as I said earlier, the rest of the SUNY system doesn't have anything like this, there's a lot of opportunities for expansion here, really across SUNY, and these are things that we're open to. We have a number of potential partners at many of the SUNY institutes, and we can start to think about SUNY-wide trials, which would be really impactful. So it's great that we have something that could have a major role in SUNY's research mission as a whole. And then we'd be open, of course, to offering other clinical trial-oriented services, given that there's a need and resources available. I do want to talk a little bit about training and mentoring. Obviously, if you want to be an institute that does a lot of clinical trials, you need to support the researchers in terms of clinical trial know-how. So to do that, we're offering something called Clinical Trial Mini Course this fall. This is sponsored not only by this new office, but by the CTSI Bird and the Workforce Development Corps, which has been a great partner to us. The goal of this workshop is not to try to turn you into a statistician or a data manager. This is to turn the audience into more educated consumers of clinical trial methodology. So the audience we have in mind are researchers and staff, and of course, students would also be interested in this as well. But really what we're trying to emphasize here is the practical aspect of conducting trials. So we're really focusing on people that are actually going to do this in the real world. You can see the schedule that we have. What I've done is I broke it into a fall and a spring schedule. The spring schedule is more of an intro. So if you're a newbie to trials, or maybe you're an established researcher and you want to kind of brush up on some concepts, then the fall series is going to be good to attend, where we're going to talk about kind of the basics of design and analysis. We'll talk about different randomization techniques. We have a talk just on pilot trials, because a lot of pilot trials happen here at UB. And we've always been of the mindset that I think a lot of people don't understand that even though a pilot trial might be a small number of subjects, there's a lot of statistical issues that you need to think through, because you're trying to set yourself up for the bigger definitive trial. And then we'll talk about clinical trial data management concepts as well. In the spring, we're going to do more of an advanced series, where we start to talk about advanced parallel group design. We start to talk about crossover trials, step wedge trials, because we do a lot of those nowadays, and pragmatic trials, adaptive designs, and trials based on time to event outcomes. Again, the mathematical level is going to be kept at a very low level. because with a lot of these concepts, especially the spring series, there's a lot of math in the background, but we're trying to emphasize the practical aspect of using this methodology. And again, this is designed really more for the clinical audience. And then we also see this as a nice training ground for mentoring. We'll be mentoring a number of students in the unit. That's of course going to enhance our reputation of the reputation of our academic programs. And this will also set us up for training grants. So we are thinking about that down the line, that we can use this as a basis for writing training grants as well. The way that we have this structured right now is we have two basic tracks for mentoring. We have a statistical track and a data management track. And I won't go through all the details in that table on the left that you see there, but you can see the sort of activities that the mentees will go through, who their mentors would be. With all of our mentees, they're going to go through training and best practices. We're going to financially support them in terms of the SAS Global Certification Program. There'll be opportunities for cross-disciplinary learning, leadership development. They'll be able to obviously participate in publication opportunities and grant writing. And then, of course, we're going to emphasize training in communication and presentation skills, which is really important for any kind of quantitative scientist. Just to wrap it up now, I just want to kind of mention the bird a little bit, because a lot of you might say, well, I don't have a big trial, right? And I have some, I'm kind of more at the earlier phase. Do I have support available to me? And the answer is yes. So you had the CTSI bird, which I also direct. The bird is really a centralized hub for clinical or for study design and statistical analysis know-how here in the Buffalo Translational Consortium. We have a really wide variety of expertise. We operate according to two aims. One is around consultation and collaboration, and the other is around mentoring, pathway development, and training. This is the bird. This is something that spans 2 institutes and three departments. All of the members are formally trained in bird-related fields and have a wide expertise and experience in conducting high-level science. There's a lot of support initiatives that we have through the bird. We have a bird consulting lab, which gives a free 10 hours, which is very unusual. You won't see that at other institutes. We have something called Research on a Napkin, which is a mechanism for supporting and giving advice to people writing pilot studies. And then, of course, we do a lot of interdisciplinary grant development with different individuals here on campus. So to wrap it up here, in terms of working with the office and some relevant contact information, even though the focus of the office is on large trials and trial-based centers, if you're interested in trials, I'm definitely interested in talking with you. Even if I can't help you, I can usually point you in the right direction to get the assistance that you might need to put together a really strong team as you apply for grants. What I would ask, especially if you're collaborating with us, is that you send me the RFA that you're responding to, a timeline for submission, any kind of tentative aims, outcomes, and eligibility criteria that you're thinking about at that point in time, and then any kind of tentative information you might have on feasibility. I'm going to ask that you contact me in 2 months in advance, and that doesn't mean that it's going to take a while for us to meet. If you email me today, I can probably meet with you tomorrow or Friday. It's really about having enough time to develop the plan that you need for this trial. There's so many moving parts. If you want to have a strong grant application, then we need the time to work on it, to work with you. In terms of my contact information, you can see my email there. Don't feel you need to write it down. I'm easy to find. I'm faculty in the Department of Biostatistics. Another key contact is Doug Lansittle, who's our chair in the Department of Biostatistics. And he can also help you in terms of finding collaborators in the statistical realm. And he's giving the thumbs up there on the screen. And he has a lot of other plans to help expand the sort of services that you'll have access to here on campus, which is really, really exciting. So thank you for your attention, and then we'll take any questions that you might have.
Well, Dr. Wilding, thank you to you and Austin for your great presentation today. And as we typically do with this series, we record the sessions and then we'll ask you for your slide deck as well, so that we can provide both as a resource to anybody who might not have been able to make it today or who wants to follow up. Since we do have another 8 minutes, Dr. Lansittle, This all initiated before you came to the university. I know that you and Greg are working very closely together on a number of different things. Did you want to take a minute to say hello?
Sure, I'd love to. And thank you, Greg and Austin, for a great talk. My name's Doug Lance. I started here February as the chair of biostatistics. And I just want to make a couple of comments. So I think both Greg and Austin did a great job of outlining the different issues and really showing the complexity. I will note that one comment, I don't work on nearly as many trials as Greg does. I'm nowhere near 200, maybe near 50, but one comment that I get either with trials or multi-site observational studies is, wow, that's really expensive. I don't usually, support all those statisticians. And I think what Greg and Austin showed is that it is a very complex set of processes that we have to deal with data management, with statistics. And we're really trying to do our best to do the best science. And to do that, sometimes it does take a team. It takes a lot of effort. So I think Greg and Austin showed that really well today. I'll just say also, so Greg and I talk frequently, and we will be working together even more closely as we get into past the rush at the beginning of the fall, and we're working to build in other structures with collaborative research, with data coordination activities. This mechanism, and I want to thank Dr. Bashir for her support, and also thank you, Jill, for all the work you've done with setting this up. We will be working on our structures that support other kinds of data coordination efforts. So again, I'm really excited. Greg and I are intertwined enough in our jobs that you contact either of us and we can get you to the right person. So feel free to contact me also.
Thanks, Doug. Very much appreciate it. Well, with that said, thanks as well to Dean Jean, who's on the call as the Dean of the School of Public Health. Biostats really has been a strength of the university under her leadership. And so we're super excited that some of these opportunities are coming up and we're bringing them to fruition to hopefully benefit all of you in your units. So with that said, Dr. Wilding, thank you again for the presentation and for all the work that you and Austin are doing. And we hope to be announcing the newest member of your team, hopefully in the next few days or within the next week or so. Any questions, feel free to direct them to Dr. Wilding, or if it's in regards to anything else, feel free to send them to my office as well.